Today we are speaking about two RNA-containing families. Members of this family medically important because they can cause severe infectional diseases with high percentage of mortality and disability. Picornaviridae family named like picorna because of small size of viruses particular. Picornaviridae family contain three genera: enterovirus, rhinovirus, and cardiovirus. Genus Enterovirus contain several members, actually poliovirus and three groups of viruses: Coxsackie A, Coxsackie B, and Enterovirus. Poliovirus can cause childhood disease, poliomyelitis. Coxsackie A, B groups of viruses can cause different diseases like myositis, myocarditis, aseptic meningitis, and actually poliomyelitis-like diseases. Enteroviruses is group of viruses with this unusual name because of one bright biological feature. These viruses don't have target tissue or target organs, and because of that, this group of viruses named like an orphans. So enterovirus, it means entero cytopathogenic human orphans viruses, and these viruses can cause aseptic meningitis, enteritis, and other diseases. And in this genus, you can see in this list, you can see one more virus. It's Enterovirus 72. This number 72, it means serotype, serological variant. And this serological variant is hepatitis A. Actually, all these viruses, viruses, have different serological variants, serotypes, and the numbers of these serotypes different accordingly to group or to virus. Biological properties of picornaviruses. In the smallest viruses, only 22 to 30 nanometers, icosahedral capsid, non-enveloped, so naked viruses, single-stranded and positive type of RNA. Stable cultivation in usual level of temperature, 37 degrees, and replication in cytoplasm because of containing exactly RNA, because of RNA gene. In diagram on this slide you can see morphology of this virus. It's quite simple morphology. Again, non-enveloped, RNA-containing virus, icosahedral, and you can see there are three serotypes of poliovirus, and these three serotypes of virus because of presence of different type of proteins in capsid of this virus. This slide contains photo of scanning microscopy and electronic microscopy of poliomyelitis virus. Poliovirus can be cultivated on kidney cells of green African monkeys' tissue and undeployed humans' cells tissue. After cultivation, or like a result of cultivation of this virus, its creation of granules in infected cells and whole destruction of tissue. And specific picture of cytopathogenic effect can be seen on right photo, where visible results of cultivation of polioviruses like a whole damaging of nervous tissue in anterior horn cells. Poliovirus can cause childhood disease. It's acute infection, and this infection lead to damaging of spinal cord and as a result neuromuscular paralysis. Epidemiology. It's only humans being infection. Source of infection apparent and subclinical patients. Incubation period from 7 to 14 days, but can be shorter, can be longer, from 3 to 35 days. Transmission by different ways. First, it's fecal-oral route and inhalation, by inhalation. Fecal-oral mechanism is because of poor hygiene. It's because of injection of contaminated food or polluted water, or by direct contact between infected and non-infected person. Pathogenesis of poliomyelitis. Virus get through the mouth to the throat and intestinal mucosa. The virus then multiplies in the tonsils. Small numbers of viruses escape to the regional lymph nodes and blood, and the virus are further amplified and cross into central nerve cells of the spinal column and central nervous system. And last stage, the intestine actively sheds viruses. Clinical manifestation. Clinical manifestation of polio depends on type of infection. Can be inapparent infection and can be clinical poliomyelitis. And clinical poliomyelitis can be like an abortive polio, it's minor illness, and can be clinical poliomyelitis with involvement of central nervous system. And again, this mild illness has two variants: paralytic polio, it can be spinal polio only, or bulbar polio, or can be complex type like bulb spinal polio, and of course it's the severest variant of the diseases, and can be non-paralytic variant of this disease. Muscle paralysis is the most important, the most prominent symptom, can sometimes result in skeletal deformities and tightening in joints and movement disability, and it's one of the factors that can lead to invalidization of victims of poliomyelitis. Immunity. After poliomyelitis, usually is active immunity. It's lifelong but type-specific immunity. Mechanism local and humoral, and the basis of local immunity is immunoglobulin A secretory that are present in the tonsils and gastrointestinal tract, and exactly this type of immunoglobulin antibodies exactly this type are able to block virus replication. And humoral immunity by creation immunoglobulin M rapid and immunoglobulin G can prevent the spread of virus to the central nervous system and to motor neurons. Passive immunity is immunity that can be given from the mother to babies by giving immunoglobulin G, but the period of working of this type of immunity only 2-3 months. Poliomyelitis can be diagnosed by two ways. First way is by detection of exactly infectional agent, I mean antigen, and second way is by serodiagnosy, by detection of immunoglobulins that are secreted in case with infectional disease. Definitive diagnosis is made by isolation of the virus from stool, cerebrospinal fluid, or a pharyngeal secretion. It means using of gold standard method. It means using of virological method. After inoculation, material that are listed on the tissue culture, and after the period cultivation, detection of presence of virus, and it means providing indication of virus. Indication of poliovirus can be by detecting cytopathological effect, like detection inclusion bodies in the host cell and by destroying of these cells. And next stage for identification is using of reaction neutralization. And one more variant, one more way for diagnosis of polio, it's using serodiagnosy. Serodiagnosy, it means detecting of presence immunoglobulins antibodies in patient's serum. And this method can be made by ELISA, immunoferment analysis, and CFT. On this slide you can see exactly CPE, cytopathological effect, after cultivation of poliovirus on culture tissue horn cells of monkey. And right photo, it's exactly CPE when visible result like a destroying of anterior horn cells. Prevention of polio. For prevention of poliomyelitis usually use or can be used two types of vaccine. First type is inactivated poliovirus vaccine. It's Salk vaccine is the author of this vaccine. And one more type, it's a live oral polioviruses vaccine that was created by scientists Sabin. Both these variants of vaccine preparated from animal cell culture and usually contain all three serotypes of polioviruses, and it's important because immunity against the poliovirus, poliomyelitis depends on serotype of virus. Most countries use oral poliovirus vaccine, a live vaccine, because its ability to induce local immunity and its huge protection against this virus. And it's a little bit cheaper variant of vaccine, but one more big plus of this type vaccine is possibility to create collective immunity. One more big group of diseases, non-polio diseases, that can be caused by group of viruses Coxsackie A, Coxsackie B, and ECHO viruses. Actually, all these viruses belong to genus Enteroviruses and can cause poliomyelitis-like diseases. But usually it's more light variants of diseases, but again, it's in the biggest part of cases it's childhood diseases, but quite similar to poliomyelitis infection in epidemiology and infectious characteristics. This subgroup of the enteroviruses can cause a variety of illnesses in human beings, and it's absolutely different, symptomatically absolutely different diseases. It can be aseptic meningitis, herpes angina, or unusual interesting disease like hand, foot, and mouth disease. It can be myocarditis, pericarditis, common cold, so a lot of different variants of disease according to this. Group A viruses produce widespread myositis in the skeletal muscles of newborn mice, resulting in flaccid paralysis without other observable lesion. Group B viruses may produce specificity effect in cycling mice, focal myositis, encephalitis. Some B strains also produce pancreatitis, myocarditis, endocarditis, and hepatitis in both suckling and adult mice. Specific types of enteroviruses infection are either subclinical or fall into the category of undifferentiated febrile illness, characterized by main symptoms like fever, malaise, myalgia. Symptoms are usually mild and last only a few days. The initial phase of infection is intestinal, after which viruses enter the lymph and blood and disseminated to other organs. The outcome of this infection largely depends on organ affected. And complications like bronchitis, bronchiolitis, croup, and pneumonia are complications that more typical for children. All ages, however, are susceptible to the common cold syndrome of enteroviruses. Pleurodenia is an acute disease characterized by recurrent sharp, sudden intercostal or abdominal pain accompanied by fever and sore throat. Lab diagnosis in case with Coxsackie group's viruses can be done by biological method. Biological model is newborn mice, and one more opportunity is gamma agglutination reaction. Initially, ECHA viruses were distinguished from Coxsackie viruses by their failure to produce pathologic changes in newborn mice. The second part of topic is Rhabdoviridae. Member of Rhabdoviridae family, it's rabies virus that cause infectional disease rabies. Rabies virus belongs to genus Lissavirus. It's enveloped RNA-containing virus that has unusual morphology. This virus has bullet-like appearance, pseudocapsid, and spikes on this pseudocapsid. Epidemiology. Rabies is a slow, infectional, progressive zoonotic disease characterized by a fatal meningoencephalitis. The primary wild reservoir of the virus are wild mammals such as canines, badgers, cats, and bats that can spread the infections to domestic dogs and cats. Both wild and domestic mammals can spread the disease to humans through bites, scratches, and inhalation of droplets of saliva. Clinic of rabies. The incubational period of rabies is from 1 to 2 months or more, depending upon the wound site and the inoculation dose. The incubation period is shorter in facial, scalp, or neck wound because of greater proximity to the brain. The prodromal phase begins with fever, nausea, vomiting, headache, and other non-specific symptoms. In the form of rabies termed furious, the first acute sign of neurological involvement are periods of agitation, disorientation, seizures, and twitching. Spasms in neck and pharyngeal muscles lead to severe pain upon swallowing. As a result, attempts to swallow or even the sight of liquids bring on hydrophobia, fear of water. Throughout this phase, the patient is fully coherent and alert. With the dominant form of rabies, a patient is not hyperactive but paralyzed, disoriented, and stuporous. Ultimately, both forms progress to the coma phase, resulting in death from cardiac or respiratory arrest. Rabies can be diagnosed by symptoms, or often the disease is diagnosed at autopsy, and criteria indicative of rabies are intracellular inclusions, its name Negri body, in nervous tissue, identification of rabies virus in saliva or brain tissue, and demonstration of rabies virus antigens in specimens of brain, serum, cerebrospinal fluid, or cornea using immunofluorescent method. On this slide, in the right part of this slide, you can see inclusion bodies exactly in host. For prevention of rabies have to be used complex immunization it means using one-time passive and active immunization. Passive immunization by using human rabies immunoglobulin, and active immunization by human diploid cell vaccine.